So the answer is it doesn't at all. There is definitely two parallel lines. One is the clinical trials, and one is the basic biology and tumor development, like genomics and functional studies, and they barely contact one another. And there are many reasons. The most is that it's kind of like easier probably for trials and for clinicians to treat patients in the same way. But we start to understand that there are certain genomic features, for example, 1114, that are particularly sensible to certain drugs like venetoclax. But again, not all 1114 are sensible to venetoclax. For example, the one with 1Q, due to the upregulation of MCL1, are probably less sensitive. And other tumors are particularly high risk. For example, the concept of high risk is keep changing, but in most of the clinical trial, we're still using a concept of high risk developed like 10 years ago. And that has been changed a lot. There are a lot of new features, and there are a lot of paradigms that have been introduced in the meantime that are completely disregarded. So I think the main challenge that I see is the integration of these two words and applying proper and a comprehensive investigation on clinical trials, because that's really the only way. And if you look, early experience from us and from other groups, it's clearly that multiple myeloma has such a massive heterogeneity that the power of the clinical trial, the current phase 3 clinical trial, for example, is probably not enough to really understand or to do subgroups analysis. And is another challenge, how we can create large repositories of, patients treated with different ways to identify and answer the question like, who should get transplant, who should not get transplant, who should get quadruplets or triplets, or what's the outcome of these patients? Also, who are the real high risk? Most of the clinical trials are on young, frail, young, fit patients, and there is a large fraction of patients that are frail, old, living in a rural area, and they are not even coming to an NCI cancer center. And so these are important considerations that I think we need to take into account when we discuss about clinical trial. Regarding the cutting edge technology, we are living in an era where there are too many technologies, and there is a kind of and most of these technologies, instead of being competition, should be seen as complementary. What whole genome sequencing can inform us, bulk, for example, is highly complementary, for example, with single cell RNA, or epigenomics, the same. So there are all these components, and still genomics needs functional validation. So we still need good functional model, like mouse model, like cell lines that properly represent the myeloma, organoids. So all these aspects needs to work kind of together in integration of this experience, and not like it's better single cell or it's better whole genome sequencing. It's probably better the combination of both on clinical trial, possibly, maybe more than one clinical trial. So you can really get a sense, an idea of how we are doing across the different groups of patients, how we can develop personalized approaches, right? All the risk in myeloma so far is based on relative risk. You are an ISS 3. What does it mean to the patient that is high risk? And then the patient is in remission for 5 years, and he still thinks he's high risk. So I don't think that patient is high risk. I think that that's a mistake from the ISS that we know is not accurate 100%. And so we also need to revise all the classification of high risk and to better understand our data. And I think it's not impossible, but I feel that it's a big conceptual effort and shift, and a lot of people are not still thinking like that. But if we really want to develop personalized treatment, that's really the only way